Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Accelerating CHO Process Optimization for Faster Scale Up Readiness. My name is Aisha Rashid, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab there. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank Nova Biomedical, who developed the content for this presentation. Advanced Instruments and Nova Biomedical are now doing business under the same brand, bringing together decades of expertise in analytical instrumentation, R&D, and global customer support. BioProfile Flex 2 is a foundational part of Nova Biomedical's integrated analytical workflow, enabling high-frequency, multi-parameter cell culture analysis within a single automated platform. Supporting applications from process development through manufacturing, Flex 2 helps generate dense, consistent process data that strengthens decision making and accelerates scale up readiness. And now it's a pleasure for me to introduce our speakers for today's event. Shweta Nair is the Director of Bioprocessing at Nova Biomedical, with over a decade of experience in the biopharmaceutical and life sciences industries. Since joining Nova in 2019, she has led the development and launch of innovative analytical technologies that improve accuracy, reliability, and data integrity across the bioprocessing workflow. Shweta drives product and business strategy through strong customer partnerships and a deep understanding of evolving industry needs. A collaborative leader, she's committed to building high performing, cross functional teams and translating customer insights into impactful solutions. Prior to Nova Biomedical, she worked in global roles at MilliporeSigma, managing laboratory equipment in biopharma and research applications. Dr. Achinta Bordaloy is a bioprocessing scientist and educator specializing in upstream process development, high throughput analytics, and biomanufacturing workforce training. At the University of Oklahoma Bioprocessing Core Facility, he has helped establish integrated platforms that support translational research, industry collaboration, and hands on training in modern biomanufacturing. His work focuses on leveraging advanced high throughput systems to accelerate CHO cell culture process optimization and applying bioprocess engineering strategies to improve biologics manufacturing. Dr. Bordaloy brings diverse international experience across leading laboratories and bioeconomy initiatives in India, New Zealand, South Africa, and the United States. So, without further ado, now I'd like to hand over the mic to our speakers, beginning with Shweta. You may begin when ready.
Thank you, Aisha, for the warm introduction. And hello, everyone. My name is Shweta Nair, and it's a pleasure to speak with all of you about the Flex 2 cell culture analyzer and how it helps accelerating CHO process optimization for fast scale-up readiness. I'd like to start with a brief overview of Nova Biomedical. We're a global life sciences company dedicated to improving patient outcomes by increasing productivity and ensuring quality across regulated laboratory environments, spanning bioprocessing, clinical diagnostics, and food and beverage. At our core, we are a partner to biopharma manufacturers focused on improving productivity, ensuring data integrity, and enabling consistent product quality across highly regulated cell culture and process development environments. Our customers really rely on us to simplify complex workflows, whether it's delivering rapid, high quality cell culture analytics with bioprofile, enabling precise media and buffer controls through our osmolality platform, ensuring accuracy and compliance in liquid handling with our Artel portfolio, or accelerating cell line development with Solenta platform. The value we bring is speed, reliability, and confidence in critical process decisions from early development through commercial manufacturing. What differentiates Nova is the combination of deep application expertise, robust instrumentation, and a global customer support network. We design our solutions around real process challenges, helping teams reduce variability, improve throughput, and ultimately accelerate their time to market for life-changing therapies. With that context, let me walk you through how we got here. Nova pioneered whole blood biosensors in 1979 and launched the first BioProfile Flex 2 cell culture analyzer in 2016. Advanced Instruments commercialized freezing point depression technology for osmolality testing back in 1955 and later expanded into cell line development and liquid handling through acquisitions of Solentum, Cell Scientific, and Artel. In 2025, last year, these two organizations came together to form a unified life sciences company, combining more than 120 years of expertise in biosensors, osmolality, automation, and bioprocessing. That foundation is really what powers the bioprofile portfolio and the reliable day-to-day cell culture monitoring we'll be discussing today. This slide gives you a bird's eye view of the markets that we serve and the breadth of our technology portfolio. Across the bioprocessing, we support customers throughout the entire workflow, from cell line development and clone selection with Solentum to osmolality testing with Osmotech, liquid handling verification with Artel, and routine cell culture analysis with Bioprofile. Today, though, we're zeroing in on the bioprofile portfolio, specifically the Flex 2, which is purpose-built for comprehensive real-time cell culture monitoring. And let me walk you through the Flex 2 in a bit more detail. The Bioprofile Flex 2 is our flagship multi-parameter cell culture analyzer. It delivers a full 16-parameter test menu covering chemistry, pH, dissolved gases, cell density viability, and osmolality, all from just 275 microliters of sample in approximately 4.5 minutes. A few things that set us apart is the comprehensive range and the modular design, so you can add on as you grow. The flexible sampling, so you are really able to sample from either a manual, a single sample, a 24 position load and go tray, or even 96 well plate for a higher throughput workflow. And the most popular as well that we're seeing an increased demand for is online sampling, where you have the ability to connect a Flex 2 to up to 10 bioreactors. And the Flex 2 is able to automatically pull samples from them and test them for this very broad range of critical analytes. Another key feature is the fact that we're able to automatically do cell counting with no manual dilutions required for up to 160 million cells per ml. We have a 48 position walkaway osmometer. We have automated QCs on board and microsensor technology, card technology, which is completely maintenance free and can be replaced in seconds, which keeps your uptime high. The instrument's also designed to really work in both process development and manufacturing, so it's built for GMP and supports 21 CFR Part 11 compliance. Now, where this really comes to life for today's discussion is when you integrate it directly with a high throughput bioreactor platform. And here, the integration that makes the case study you're about to see possible is the Bioprofile Flex 2, where it connects directly to a Sartorius AMBER 250 system through the external sampling module. This enables for a fully walk-away automated sampling. This is the integration that makes the case study you're about to see possible. The BioProfile Flex 2 connects directly to the Sartorius AMBER 250 system through the external sampling module, enabling fully walk-away automated sampling. You get a complete 16 parameter analysis in 6 to 7 minute cycle time, just using 400, anywhere between 400 to 500 microliters of sample. The results are transmitted in real time directly back to the AMBER 250 software for feedback, process control, with OPC connectivity for LIMS and historian integration. The system delivers zero carryover through automated fluid path cleaning and deproteinization after every sample, and reagent capacity supporting unattended operation for over a week. And this instrument also supports 21 CFR Part 11 compliance with automated daily QC, so it's GMP ready out of the box. This is the exact setup that Dr. Bertollet used in the study he's about to walk you through, where the AMBER 250 and the Flex 2 combined with design of experiments were used to accelerate CHO process optimization and achieve scale-up readiness. With that, I'd like to hand over to Dr. Bertollet. Thank you.
Hello everyone, and thank you for joining today's webinar titled Accelerating CHO Process Optimization for Faster Scale-Up Readiness. My name is Achinta Bertollet, and I'll be discussing how integrated high-throughput bioprocess development tools can significantly accelerate CHO cell process optimization for monoclonal antibody production. In today's presentation, I'll be focusing on how this study combined automated AMBER 250 high-throughput bioreactors, real-time analytics using the Nova BioProfile Flex 2, and design of experiments or DOE to create a rapid and scalable workflow for upstream process optimization. So we walk through the various steps, rationale, workflow, outcomes, results, and concluded implications for future biologics manufacturing. Here's the agenda for today's session. We will begin with some background and current industry context surrounding biologics manufacturing. Next, we will discuss why CHO cells remain the dominant host platform for therapeutics protein production. We will then move into CHO process optimization workflows and discuss how high-throughput bioreactors, real-time process analytics, and statistical experimental design can accelerate development timelines. Following that, we will present a case study demonstrating how these technologies were integrated to optimize monoclonal antibody production. Finally, we'll summarize the key takeaways and discuss future directions in data-driven bioprocess development. So the demand to accelerate biologics such as monoclonal antibody process development timeline using different host cells to bring therapies to patients sooner is gaining momentum. The biologics industry continues to experience tremendous growth, with the global market projected to exceed $975 billion by 2030. With the surge in demand for biologics, there is increasing pressure on biopharmaceutical manufacturers to reduce process development timelines, lower manufacturing costs, and improve process robustness while maintaining product quality and regulatory compliance. Mammalian expression system, particularly Chinese hamster ovary or CHO cell lines, remain the dominant platform for monoclonal antibody production and account for approximately 80% of approved biologics. However, conventional process optimization approaches are often labor-intensive, sequential, and resource demanding. To address these challenges, the industry is increasingly moving towards automated, high throughput, and data driven workflows. So, the goal of this work was to accelerate CHO process development by integrating single use, high throughput bioreactor systems such as the Ember 250 bioreactors, facilitating precise and automated control, real-time process monitoring using the Nova Bioprofile Flex 2 integrated to the system, and design of experiment methodologies. Why CHO cells? CHO cells have become the gold standard for biologics manufacturing because they provide an optimal balance between productivity, product quality, scalability, and regulatory acceptance. One major advantage is their ability to perform human-like post-translational modifications, which are essential for the efficacy and stability of many therapeutic proteins. CHO systems also have an extensive regulatory track record, which reduces development risk during clinical and commercial manufacturing. From a manufacturing perspective, CHO cells exhibit high productivity, strong genetic stability, and compatibility with large-scale suspension cell culture systems. As a result, they are routinely used for producing monoclonal antibodies, biosimilars, and a host of various recombinant therapeutic proteins. Despite these advantages, the production process in CHO cells is complex, and maintaining productivity, quality, efficiency, and consistency remains challenging. Some biopharma industry bottlenecks. So, despite advances in biologics manufacturing, the critical bottlenecks continue to slow process development. These include high development costs, long optimization timelines, process variability, and challenges associated with scale-up and reproducibility. Another key issue is the limited ability to rapidly screen and optimize multiple process conditions simultaneously using traditional bench-scale bioreactors. To overcome these limitations, the industry is increasingly adopting integrating data-driven solutions that combine automation, high-throughput experimentation, single-use systems, real-time data analytics, and advanced statistical modeling. So these technologies enable faster decision-making while improving process robustness and scalability. This slide summarizes the overall workflow used in this study. Here, a systematic mapping approach was employed to identify critical process parameters and improve process efficacy. The first step involved developing a systematic process mapping and optimization framework using design of experiments. We then used automated Ember 250 high-throughput bioreactors as a scalable high-throughput model system. The Ember platform was integrated with the Nova Bioprofile Flex 2 analyzer to enable real-time process monitoring and rapid analytical data-driven feedback. Using this integrated workflow, we evaluated critical process parameters that influence monoclonal antibody productivity and cell culture performance. Based on the design of experiment, a model was developed and the optimal conditions for higher MAP titer were predicted and further validated to confirm improvements in antibody titer and culture variability. The schematics on the right kind of illustrates the overall steps towards map production. So now let's briefly discuss the Ember 250 high throughput platform and what it entails. So this system contains 12 parallel single-use mini bioreactors, each capable of independently controlling key process parameters such as pH, dissolved oxygen, temperature, and agitation. With an operating volume of up to 250 milliliters, this allows for simultaneous experimental runs in parallel across multiple conditions using design of experiment to optimize critical process parameters. The platform is fully automated and integrated with runtime software for process control and sampling. One of the major advantages of the Ember system is its ability to function as a reliable scale-down model for larger manufacturing bioreactors. It offers better scalability and tech transfer from bench to pilot scale during early stages of the development pipeline. Because multiple conditions can be tested simultaneously, the system enables highly resource-efficient experimentation while dramatically accelerating process development timelines. The Nova Bioprofile Flex 2 served as the analytical backbone of this workflow. This automated cell culture analyzer uses microsensor technology to rapidly quantify 16 critical cell culture parameters. The system measures a broad range of analytes including glucose, glutamine, glutamate, lactate, ammonium, sodium, calcium, potassium, total cell density, viable cell density, cell viability, live cell diameter, pH, osmolality, and partial pressure of carbon dioxide and oxygen. So the ability to obtain rapid and reproducible analytical data enables real-time process monitoring and supports data-driven optimization strategies. Integration of the Flex 2 with the Ember platform significantly improved workflow efficiency and reduced analytical turnaround time. For this particular study, we used a CHO cell line, MyCHO from Atom, producing an IgG-based monoclonal antibody. Cells were cultured in XCell Advanced CHO medium, supplemented with Cellvento Modify. Both are commercially available growth and feed media. The process was operated in fed batch mode for 14 days under controlled conditions. Dissolved oxygen was maintained at 30%, pH 7, and agitation set at 330 RPM. The temperature shift from 37 degrees Celsius to 33 degrees Celsius was implemented once the viable cell density exceeded 10 million cells per ml or by day 5, whichever is earlier. Daily feeding began after this temperature shift based on the experimental design. This workflow was designed to evaluate how process variables influence cell growth, metabolism, and antibody productivity. To systematically evaluate process conditions, we applied a central composite design or CCD within a design of experiments framework. A DOE-aided approach was applied in this study to optimize the operating range of two critical process variables, seed density and feeding rate. Seeding density ranged from 0.2 to 1.4 million cells per ml, while feed rate ranged from approximately 1.2 to 3.3% vessel culture volume per day. The primary response variable was monoclonal antibody titer. Statistical modeling and response surface optimization were performed using JMP Pro 18. This approach allowed us to efficiently identify interactions between variables and determine optimal operating conditions with a minimal number of experimental runs. The table on the right shows the experimental matrix generated through the central composite design. Each Ember bioreactor represented a unique combination of seeding density and feed rate conditions. The inclusion of center points and axial points enabled robust modeling of both linear and quadratic effects within the experimental design space. This experimental structure provided the statistical power needed to evaluate parameter interactions and generate predictive response surface models. One of the strengths of this workflow was the integration of process control with real-time analytics. So the BioProfile Flex 2 was connected directly to the Ember high-throughput system, enabling rapid monitoring of metabolites and cell culture performance. Automation from Ember and real-time monitoring using Flex 2 allowed seamless control of parameters ranging from pH and glucose level and many other parameters as well. For example, target glucose and pH control levels were analyzed by Flex 2 at the desired frequency, and immediately the Ember control activates the pump feed for addition of glucose or base to maintain target levels. Additionally, CO2 gassing is activated if there is need to lower the pH rises. So this created a feedback-driven process optimization loop where real-time analytical data enhanced better control of the run and informed process understanding and decision-making as well. In addition to in-process monitoring, offline analyses were also conducted for metabolite profiling and IgG titer quantification to monitor culture performance and productivity. Together, these analytical capabilities enable comprehensive evaluation of culture performance throughout the fed batch process. Now let's review some of the results. Here we see the impact of process conditions on viable cell density on the left and cell viability on the right, shown for all 10 bioreactors designated as P1 to P10. The peak viable density occurred between day 5 and 7, with maximum densities reaching approximately 15 million cells per ml. Temperature shift to 33 degrees Celsius was applied to reactors between day 3 and 5 upon reaching a preset condition of VCD reaching 10 million cells per ml, along with studying the feeding rate at the specified percent as per the DOE table, or day 5, whichever is earlier. Reactors with higher seeding density greater than 1 million reached the 10 million mark by day 3, where the one with lower seeding density took few more days. Irrespective, the temperature shift was applied on day 5 for those who didn't reach the 10 million mark. The graph on the right illustrates the cell viability profile over the time course. Overall, viability remained above 85% across most conditions, demonstrating strong culture robustness. However, as you can see from bioreactor 10, in red dots operated at the lowest feed rate of 1.9% showed reduced viability, which dropped to 72% at harvest, indicating nutrient limitations and metabolic stress under insufficient feeding conditions. These results highlighted the importance of feed optimization for maintaining healthy and productive cultures. It also allows to potentially increase culture time to increase process efficacy if the culture stability and productivity can be maintained. this slide presents metabolite trends across the different process conditions. On the top left, the lactate profile peaks during the exponential growth phase to an average of value of approximately 1.3 grams per liter. The lactate shift was observed from day 6 to 8 towards consumption until day 12. The lactate value stabilized to around 0.1 grams per liter towards the last few days from day 10 to 14 until harvest. So this switch is considered favorable and often serves as a key metabolic tuning characteristics. However, the exact intrinsic causes driving this still needs more investigation and its impact on titer productivity. On the right, the ammonium level increased more under low feeding conditions, suggesting amino acid catabolism and nutrient stress. Although the overall profile of ammonium concentration across culture conditions did increase during the production phase, but it also correlated with the glutamate accumulation in the culture. The glutamate concentration showed a very strong correlation with feed rate, precisely due to its presence in the added feed media. That's why a statistically significant relationship was found with an R-square value of 0.9. Data not shown here for that. And the highest accumulation of ammonium in the stationary phase was accompanied with the lowest glutamate concentration, which is B10 in the red again, as can be seen in the plots for both ammonium and glutamate. It seems that at lower feed rates, the CHO cells faced an unbalanced nutrient supply that may impact the cell activities and metabolism. It also impacted the cell viability, as discussed in the previous slide. Overall, these findings demonstrate how feeding strategy directly influences metabolic behavior and culture performance. Now turning to our main response variable, which was the monoclonal antibody productivity. Final titer ranged from approximately 3.1 to 4.5 grams per liter by day 14. The highest titer was achieved at a seeding density of 1.4 million cells per ml and a feed rate of 2.25% vessel volume per day. In contrast, the low seeding density and low feed rate conditions produced titers below 3.5 grams per liter. Importantly, conditions with seeding density above 1 million cells per ml and feed rates above 2% showed more than 40% improvement in productivity. So, these findings reinforce the strong influence of nutrient delivery and initial cell density on process performance. This slide summarizes the statistical optimization model generated from the DOE analysis. Multiple regression analysis was implemented to analyze and fit the linear and quadratic equation to the experimental data set. All the estimated parameters, including the values of coefficients, interactive terms, t-ratios, and p-values for the model, were assessed for statistical significance. The figure on the right is the response surface plot showing the impact of seed density and feeding rate on the production of MABs. The response surface model showed a strong fit with an R-squared value of 0.92, indicating high predictive accuracy. Feed rate demonstrated both significant linear and quadratic effects, while seeding density showed a significant linear effect. And to supplement that, the lack of fit p-value was not significant, confirming good model reliability. So that using the desirability optimization profile in JUMP, the predicted optimal conditions were at a seeding density of 1.1 million cells per ml and a feed rate of 2.6% vessel volume per day. Under these conditions, the predicted model antibody titer was approximately 4.57 grams per liter. To validate the predictive model, duplicate confirmation runs were performed using the optimized process conditions. The measured antibody titer reached 4.41 grams per liter by day 14 and further increased to approximately 5 grams per liter by day 16. The culture variability, or rather viability, remained above 84% throughout the time course, reaching a peak VCD of approximately 14 million cells per ml. Productivity was approximately 0.33 grams per liter per day, confirming strong process performance. The culture period was extended to day 16 to further investigate the MAP production and growth parameter trends as impacted by culture time. Although the cell viability dropped to 77% at harvest on day 16, but MAP production titer increased to 5 grams per liter. So most importantly, these validation runs demonstrated excellent reproducibility and robustness of the optimized conditions. This study demonstrated that both feed rate and seeding density significantly influence CHO culture performance. Low feeding conditions resulted in nutrient stress, elevated ammonium accumulation, and reduced cell growth. The observed lactate shift following temperature reduction also highlighted important metabolic adaptations during fed batch culture. There is more room to further investigate this aspect to gain more mechanistic understanding. From a platform perspective, the AMBER 250 proved to be a highly effective scaled-down model for monoclonal antibody process development. And in parallel, the NovaFlex 2 enabled rapid analytical feedback that supported real-time data-driven optimization. Together, these technologies created an efficient workflow for accelerating upstream process development. The key takeaway from the study, and to summarize the overall findings, combination of automated bioreactor systems such as the AMBER 250 statistical modeling via DOE and the BioProfile Flex 2 enabled real-time monitoring, provided a strong tool for studying and optimizing map production using lower resources and significantly less time. It also accelerates CHO process development optimization, as CHO fed-batch is still the predominantly adopted route at commercial scale while allowing smoother tech transfer to existing manufacturing infrastructure. The optimized conditions at seeding density of 1.1 million cells per ml and a feed rate of 2.68% vessel volume per day enabled achievement of approximately 5 grams per liter of MAP titer with strong reproducibility. So the result indicated that by fine-tuning of the MAP production process variables, maintaining cell viability and productivity is attainable even beyond the traditional 14 days. Importantly, this workflow provides a scalable and resource-efficient framework for modern biologics manufacturing, and this can favorably impact screening and process development timelines and offer reliable tech transfer pathways which would eventually allow quicker access of therapeutics to the patients. these findings have several important implications for industrial bioprocess development as well. First, the high throughput automation enables faster process mapping while reducing labor and material costs. Second, DOE-driven optimization improves process reproducibility and supports more efficient identification of critical process parameters and mapping them to process efficacy and productivity. And finally, validated optimized conditions provide a strong foundation for scale-up and technology transfer into pilot and manufacturing environments. And insights from this kind of research and other empirical data can guide future research focusing on novel bioreactor and alternative strategies for process development such as intensified fed batch and perfusion cultures to further boost productivity. Now looking ahead, several opportunities exist to further enhance this workflow and R&D goes on. Further studies may focus on harnessing genetic engineering tools to control metabolic pathways and to generate more stable and productive cell lines. There's always a space for optimizing feed composition to improve both productivity and product quality attributes. Additional integration of online sensors could enable continuous real-time metabolic monitoring and closed-loop process control. There is also significant potential for implementing AI and machine learning-driven predictive modeling approaches to support adaptive and autonomous bioprocess optimization. And we can see this space growing more and more on as we move. And together, these advancements could further reduce development timelines and improve manufacturing efficacy. Here are some references, and the core results presented in this webinar has been published and can be accessed from the publisher's website using the link provided. So thank you for your attention. At this point, we would be happy to take any questions or discuss any aspects of the workflow. So thank you again for joining today's webinar.
Well, thank you very much to our fantastic speakers, Shweta and Achinta, for that fabulous presentation. Very, very insightful. And indeed, we are now heading into the Q&A portion of the webinar. So audience members, please continue sending in your questions for our speakers, and we'll try to attend to them during our Q&A session here now. I've already received some questions here, so I think we can get started with those. All right, so the first question that we have here is, pertaining to a piece presented, of course, in the presentation. Is this a clone or pool? Wondering if you have data regarding the performance of the cell line in a shake flask and seed density, feed rate, and temperature shift timing, just to compare process improvements in the Ambr 250 versus shake flask.
Yeah, it's a clone, and we did have preliminary shake flask data to kind of identify some and get some a priori data. At the shake flask, of course, we are around 2.8 to 3 grams per liter tighter. So yeah, we could improve that up to 5 in Ambr.
Thank you so much, Chinta, for that answer. Another question we have here is, what are you hearing from customers about how real-time analytics like the Flex 2 are changing their upstream workflows?
I think across the board, we're hearing from customers that real-time data that we're getting from especially these 16 parameters on the device can be very helpful in
optimizing their feed strategies, and making sure that they're making the right decisions throughout their process. This is a growing area. It's something that, as even Achinta alluded to in his presentation, there's so much more that can be understood, and so much more that needs to be understood, and so much more that we as a vendor are also trying to work with our customers closely on.
Thank you so much, Shweta, for that answer. And also, another question pertaining to that as well is, can Flex 2 also connect to an Amber 15 instrument?
Yes, so you can connect a Flex 2 to the Amber 15 system as well, and it would be able to achieve similar data set in terms of pulling the sample from the cell culture reactors on the Amber 15 to give you the full spectrum of data.
Great. And another question that we have here is, what made you land on seeding density and feed rate as your two DOE design of experiments factors over other process parameters?
I think they are two of the most actionable variables from a culture perspective to hone down on. And also, we have some a priori preliminary experimental data, which kind of suggested um, why they are important. So, from an DOE perspective, it's good to harp on the, the, the few more critical one and just kind of expand the design space at different levels and before moving on to fine tuning some of the other variables. So it's not everything at once, but just on a structured mapping. So that was the idea of kind of how we optimize and how we kind of map those critical parameters so that it gives some more room. And so that's kind of what's ongoing right now. Which we would hopefully publish soon. So we're hitting beyond 6.5 right now with our ongoing experimental, which was building on to what we did and what was presented today. So.
Great. Thank you so much, Chinta, for that answer. And the next question here is, you reached about 5 grams per liter in the validation runs. How in the validation runs, excuse me. And how confident are you that those conditions will hold up at larger scale?
Yes, I think partly I answered the question in the previous one where we were hitting beyond 6, but at the Amber level as well. We actually did some scale-up validation. So that's, I mean, Ember and the Flex integration has been validated as a scaled-down model because there are a lot of, there are few published results already where they have validated that the mini bioreactor system as a good scaled-down model because they came from 5 liters down and kind of validated that at a scale down. And I think the reverse is also possible, and we could do that at 3 liter in one of our other single-use vessels. And so it's doable. So we're fairly confident that we can move further out. And because of the geometry of how it is designed, so it is pretty scalable. So that's the good part.
Fantastic. And let's take a look at another question here. For teams looking to adopt this kind of integrated approach, what's the one piece of advice each of you would give?
Okay, I can start off with.
You should
Okay.
start off, Rajita, and I can
Okay.
join in.
Yeah, okay. I think from a process development perspective, it's again just trying to understand, not like optimize everything at once, or rather try to do it the more critical one, which had more influence on the kinetics, the growth, and the productivity, because and gain a more mechanistic understanding of how the cell line behaves, how it all behaves, and then layer it up with some of the other fine tuning, also like lactate and other stuff. That is one aspect. And I think from the analytical part, I think having that robust workflow in place is very critical so that you know that data gathering, empirical data set, and a more reliable data set, because that can then feed into more of process modeling, for
Mhm.
predictive modeling, because data is king. If we move into those integration of AI and machine learning, and that can be, Sweta can probably add on to how Flex helps us do that. Yeah.
Yeah, no, absolutely. I think for us as a vendor, we're really looking to understand the scope, how the customers are envisioning their workflow, because the analytical device such as the Flex 2 is only enabling them and providing some of that insights. And so we really want to make sure we're working with customers to understand how the instrument will be used with the Amber, how often, but also what that data is going to mean to them, sort of what like Achinta alluded to, because that's where the insight really lies for decisions that they will be making through their workflow. So at Nova Biomedical, a big asset that we believe that we're able to offer customers is our applications team that are a free of charge service solution that we have on site. That they can come on site, work to optimize, make sure the instruments are measuring the analytes, measuring the data set absolutely accurately, also help with the automation angle, so sending the data to whatever cloud-based or ELN network or whatever the customer may have set up, to then be able to orchestrate on the back end all of the insight on their side. So for us, the only advice we would give is make sure you're considering us a partner in your process. So the more you're able to give us your vision and work with us, I think the most easy the implementation and the faster the insight will be for the customer at the end of the day.
Wonderful. Fantastic. I'd like to thank our speakers once again, Shweta and Achinta, for answering great questions, by the way, for sure. And once again, for that fantastic presentation. We've now reached the end of the webinar. And if we couldn't attend to your questions, the team at Nova Biomedical may follow up with you. Also, just wanted to bring to your attention that for those who are interested in hearing more from Nova Biomedical, you can register for their next upcoming webinar entitled Scaling Cell Line Development, Enabling IND-Ready GMP-Compatible Workflows, which is scheduled for Tuesday, June the 2nd. And there's a second broadcast of the webinar as well on Wednesday, June the 3rd, just the next day. So I'm going to send links to these broadcasts, and you may be able to. I do encourage you to register, and so that you can stay tuned and tune in for those broadcasts of that webinar. Just sent in the first one there, and I'm going to send the link to the second one there. There we go. All right. And as I mentioned, if we couldn't attend to your questions, the team at Nova Biomedical may follow up with you. Or if you have any further questions, you may also direct them to the email addresses displayed on your screen. I'd like to thank everyone once again for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. In addition, I'm going to share another link in the chat box, and this is for today's webinar. So you can register if you haven't already, and you can share this link with your colleagues as well, so that when they register for the recording, they can view it there. As well at any time on demand. So I highly recommend that you do go ahead and share the link to today's webinar, which once again was truly very informative and insightful. All right. And for those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speakers once again, Shweta Nair and Dr. Achinta Bordoloi, for their time and insights today. And on behalf of the team here at XTalks, we'd like to thank you for your participation and attending this webinar today, which we hope you found informative. On behalf of the team here at XTalks, once again, thank you once again, and we hope to see you at the next webinar. Take care and bye for now, and have a great day, everyone. And thank you once again, Shweta and Achinta.
Thank you, Aisha.
Thank
Thank
you.
you, Xtath. Thank you,
Thank you.
everybody.
Thank you. Take care.